Hello everyone, welcome back to BMI5730. We will continue with Module 4 on proteomics. In our last lecture, we talked about what is proteome and what is proteomics. We talked about what we measure in proteomics and in what application scenarios we can apply proteomics technologies. Today, we will dive deeper into the data types and analysis. Here is the scope of proteomics research. Just to refresh what we have learned by now, what is the question you want to answer? For example, how does the protein abundance change in response to some stimulus? You may be interested in a specific protein or a group of proteins, or you are interested in all proteins that are detectable in your samples. You may ask, how does the protein modification status change in response to some stimulus? Do we detect different proteins in two different samples? Are different proteins secreted from the same tissue under different conditions? How do the proteins interact with each other in different conditions, etc.? There are some keywords: identification, quantification, modification, structure, interactions, function, and so on. Now, for the proteins that we study, we want to know their identity and quantity. We may monitor the proteins using mass spectrometry-based methods. Well, these are not the only type of methods we can use, although they are popular. In this example, you can see that there are more than 5,000 proteins detected in the serum proteome. The y-axis is the log 10 concentration, the normal range of the abundances of the proteins. You can see that the abundances of proteins vary. Some proteins are highly expressed, while others are not. Other than the protein abundance, in some studies, we're interested in post-translational modifications. This figure shows the biochemical processes of several modification forms, such as phosphorylation, ubiquitination, acetylation, methylation, and proline hydroxylation. We are not going into detail in the lecture, but you are welcome to read the paper listed on the slide after the class. Take phosphorylation, for example. We may use the Silac methods to detect modifications after the phosphopeptide enrichment process in the sample preparation. Silac stands for stable isotope labeling with amino acids in cell culture. It is a mass spectrometry-based technique. It detects differences in protein abundance among samples using non-radioactive isotopic labeling. Silac is popular in quantitative proteomics. Various post-translational modifications represent protein heterogeneity. There is much larger number of variant modification spots compared to the common proteins they represent. For example, for the proteome of H. influenzae, there are around 1,500 modification sites, but they only reside on around 500 different proteins. H. influenzae is a bacteria, not a virus that causes flu. More than 200 post-translational modification forms have been identified. A single protein may carry several modifications. Modified proteins show different properties compared to unmodified counterparts. In most cases, we do not know the origin or the biological significance of the observed heterogeneities, and we want to use proteomics techniques to help solve these unknowns. In classical proteomics methods, proteins are separated on a two-dimensional gel by their physicochemical properties. Proteins are first separated based on their charge using IEF, then further separated based on their size, as in SDS page. Then gels are stained, and the protein spots of interest are excised from the gel. Then trypsin digested. One such spot is shown with a red circle in the figure. After trypsin digestion, Peptides are extracted and sequenced using tandem mass spectrometry. We also call tandem mass spectrometry MSMS. Proteins with different physical-chemical properties, including those with the same backbone sequence but with different modifications, were located on different spots of the two-dimensional gel. From MSMS experiments, we can know the amino acid sequences and the modifications of the proteins in the sample. Such proteomics methods can help us investigate the heterogeneity of proteins. Here in the rectangle of the MS peaks and the MSMS peaks, we call them the mass spectra. Let me read the description. It is very good. For the generation of MS peaks, peptides are ionized and resolved on the basis of M/Z. That's the mass-to-charge ratio. From the resulting mass spectrum, parent ions, the red peak in the figure, are selected for further MSMS analysis. Let's look at the peaks of MSMS in the top right corner. Differences in fragments ion masses in the MSMS spectra, see the double-headed arrow, can be used to determine the sequences of the peptides. This process sounds complex. However, luckily, we usually use the existing computational tools to determine the sequences from mass spectrometry raw data, usually by using database search. We will teach you how to do it in the next lecture. I just briefly talked about how MS spectra are generated. The figure on this slide shows how a mass spectrometer works. When the sample, which is the ion source, is injected into the machine, precursor ions are selected and isolated. Then collision-induced dissociation results in fragmentation. Finally, product ions are characterized with a second mass analyzer. The machine will record the signal output as mass spectra. For more technical details, I have provided 2 YouTube links on this slide. I will also provide the same links on Carmen. For mass spectra, some points we want to mention. Proteins commonly consist of 20 different types of amino acids with different physical-chemical properties. While there are exceptions in some species, the amino acids composition are different than ours. Here I provide you the link to amino acid browser to check the atom nomenclature, structure, properties, abbreviations for the 20 common L amino acids. Some of them are more similar, while others are more dissimilar. Different peptides produce different spectra. Because of this unique relationship, we can use the spectrum of a peptide to determine its sequence. This is also the reason why we want to use database search to find the most possible sequences with a spectrum that matches what we observed in the MSMS experiments. After we get the amino acid sequences, we can try to infer the protein structures from the sequences. In this example, despite being 59% identical, the 2 protein sequences fold to 2 different structures. You may read the reference for more details if you are interested. Computationally, there are existing tools for us to predict the protein structure from sequence. You can check the website of CASP. CASP stands for the Critical Assessment of Techniques for Protein Structure Prediction. It is a competition, and the website provides information about winner structure prediction algorithms over the years. You may check iTester, one of the algorithms. It is available from Yang Zhang Lab at the University of Michigan. We can go even further from the sequence. We can predict protein-protein interactions, or say PPIs, from the amino acid sequences of 2 proteins. This is a slide with a lot of minifigures of protein structures. We just want to say that our hypothesis is the protein structure is determined by its sequence and modifications. From the sequence, the protein's physicochemical properties are inherited. And from the protein structures, we can predict which proteins could have interaction and how the interaction happens. One of the PPI prediction tools is Struct2Net. Its server is maintained by Dr. Barney Burgess' group at MIT. Based on the introduction on their webpage, the Struct2Net server makes structure-based computational predictions of protein-protein interactions, PPIs. The input to Struct2Net is either 1 or 2 amino acid sequences in FASTA format. The output gives a list of interactors if 1 sequence is provided, and an interaction prediction if 2 sequences are provided. You may use Struct2Net as one option when you need to predict PPIs from the sequences. We have just talked about classical proteomics methods. Mass spectrometry-based methods are popular, but they are not the only options. Gel-based techniques, they are old school but still widely used and effective in the right situation. Let's revisit the figure of classical proteomics methods. We can use a 2-dimensional gel to separate proteins and excise the spot of the protein from the gel, and proceed to further mass spectrometry experiments. Well, you may see that the operation is low throughput, since we need to excise the spot of interest one by one. Then people moved to more new school techniques without 2-dimensional gels. For example, mass spectrometry-based methods such as LC-MS-MS. Here, LC stands for liquid chromatography. MS-MS stands for tandem mass spectrometry. In LC-MS-MS experiments, we don't need to excise gels anymore. Basically, LC does the job to separate proteins. Proteins with different physical-chemical properties run out of the LC column at different times, and the process is continuous. Proteins continuously run out of the columns and feed into downstream MS-MS experiments. These techniques are very popular. They're quantitative and high throughput. If you have heard about OSU Proteomics Shared Resource in the Biomedical Research Tower, they have mass spectrometers, and they frequently run MS-based platforms. The third category is array-based methods. They're quantitative, very sensitive in detection, but limited in the scope of proteins an array can analyze. Unlike MS-based methods that monitor all the proteins in the sample, array-based methods only monitor the proteins that have designed antibodies on the array. That means if you do not have the antibody on the array for a specific protein, that protein will not be monitored. For example, RPPA, reverse phase protein lysate microarray, it has been used to generate proteomics data for TCGA, the Cancer Genome Atlas, for tissue samples of multiple cancer types. In summary, no matter what platform you are using and how many proteins you are monitoring, the output of a proteomics experiment would be the identification and the quantification of the proteins. The experiment tells about proteins you can detect in your sample, what modifications are there on the proteins, and what abundances the proteins have. We can also know the sequences of proteins. For classical methods using trypsin to digest input proteins, we got the sequences of the peptides from digested proteins. Here is an example of a comparative analysis from a published paper. LC-MS-MS-based comparative proteomics of floral nectars. In brief, they extracted the floral nectar protein samples, did trypsin digestion, and put the resultant peptides into LC. LC does peptide separation, and then the peptides were input into a mass spectrometer for peptide sequencing. From the mass spectrometry raw data, we can get the identification and the quantification of the peptides and proteins. And they did bioinformatic analysis comparing multiple samples. This is a Venn diagram comparing 3 different species. The experiments were conducted using nectar samples from multiple flowers from 3 different plants, so they have 3 independent biological replicates. From the analysis, they got what proteins were detected in multiple species and what proteins were only detected in 1 or 2 species. They can even compare the protein abundance using traditional differential expression analysis. In their analysis, the data were evaluated using a one-way ANOVA test. The differences between species were analyzed using a student t-test at p-value smaller than 0.05 significance level. You may think about other downstream bioinformatics analysis you can do, and we will do some practice using example data later. Thanks for watching. See you next time.